Development of bone or ossification. Bone is a special type of connective tissue, mesenchymal in origin. It develops by two methods: intramembranous ossification or intracartilaginous ossification. The basis of each process is the same, that is, the conversion or replacement of the primitive connective tissue into bone. If the process occurs directly, it is called intramembranous ossification. But if there is the formation first of a cartilaginous model, then replacement of such model by bone, the process is called intracartilaginous or endochondral ossification. Intramembranous ossification is the conversion of a membrane of mesenchymal connective tissue into bone without passing into a cartilaginous state. It occurs in flat, short, and irregular bones. Stages of intramembranous ossification. Primary center of ossification develops in the middle of the mesenchymal membrane. This center is characterized by high vascularity, so mesenchymal cells are differentiated into osteogenic cells and osteoblasts. In a vascular environment, these cells grow and differentiate to chondroblasts. Osteoprogenitor cells or osteogenic cells enlarge to form osteoblasts, which secrete bone matrix and deposit calcium salts due to their content of alkaline phosphatase, thus forming the future embryonic bone, formed of interlacing bars or trabeculae radiating from the center of the membrane to the periphery, extending in a radial manner outwards. Osteoblasts remain connected together by their long cytoplasmic processes, and once they are imprisoned in calcified matrix, they are changed into flat, mature osteocytes. The process of ossification occurs in several fossae, resulting in multiple bony bars that branch and anastomose freely and are separated by spaces filled with mesenchyme. This process continues until the entire mesenchymal membrane is converted into the future cancellous bone. Mesenchymal connective tissue present between the bony bars forms the bone marrow cells and endosteum, and that on the surface forms periosteum. Embryonic bone formed in this way is immature cancellous bone. This is later on replaced by mature cancellous bone, which may remain as such as in the dipolo of the skull or the center of flat bones, or changes to compact bone as in long bones by continuous deposition of new bony lamellae on the walls of the marrow spaces, which gradually become narrowed to form the future Haversian canals and Haversian systems. Intracartilaginous ossification or endochondral ossification is the replacement of a cartilaginous model of hyaline cartilage by bone. This occurs in long bones and in the outer part of short bones. Diagram for development of long bone. In the center of the shaft or diaphysis of a cartilaginous model, the chondrocytes hypertrophied deposited calcium salts. As a result of that, they cut off their nourishment as they depend on diffusion, degenerate, and die. The overlying vascular perichondrium assumes an osteogenic function and produces a layer of bone, periosteal collar, around the altered cartilage by intermembranous ossification to maintain the strength of the shaft. The perichondrium has become periosteum, invades the degenerated cartilage, forming periosteal bud, rich in blood vessels, osteoblasts, and osteoclasts. These osteogenic cells lay down bone matrix and form the primary center of ossification in the diaphysis. The secondary ossification centers appear at the time of birth in the extremities of cartilage model, that is epiphysis, and expand in all directions, resulting in a mass of spongy bone. This process of ossification continues in both the epiphysis and diaphysis and results in complete replacement of the cartilage model with bone except in two regions. The two regions which remain cartilaginous are the articular cartilage present throughout life at the end of the bone and the epiphyseal cartilage or the epiphyseal plate joining the epiphysis to diaphysis and is responsible for growth in length of the bone, interstitial growth before adulthood, and stops in adults. Diagram of the epiphyseal plate during endochondral or intracartilaginous ossification, showing the various stages of ossification. The epiphyseal plate is the cartilaginous plate present between the epiphysis and diaphysis and is responsible for bone growth in length, interstitial growth before adulthood. Zone of resting or reserve cartilage consists of several layers of chondrocytes in their lacunae randomly distributed in the matrix. They show slow rate of growth in all directions and act as reserve cells for other zones. Zone of proliferation. Cartilage cells distal to the resting zone divide repeatedly, giving rise to a large number of flattened chondrocytes, present singly inside their lacunae, not in nests, arranged in rows, leading to increase in the length of the cartilaginous plate. Zone of hypertrophy and maturation. Chondrocytes stop mitosis, enlarge, accumulate glycogen, and become rich in phosphatase. The lacunae also widen and become separated by thin bars of matrix. Zone of calcification and cell death. The enlarged chondrocytes deposit calcium salts in the surrounding matrix, helped by alkaline phosphatase enzyme in their cytoplasm. The calcified matrix, being impermeable, cuts off nourishment to the chondrocytes, which depend on diffusion and imbibition. Chondrocytes in turn die and degenerate, leaving empty lacunae. This is followed by breaking down of the thin bars separating the lacunae, resulting in formation of irregular cavities separated by trabeculae of calcified matrix. Zone of ossification and internal reconstruction or remodeling. The overlying vascular perichondrium has become periosteum and assumes an osteogenic function. It has to compensate for the weakness of the cartilage model plate caused by death of the chondrocytes. So it forms a very thin collar of bone under the periosteal, subperiosteal bone by intermembranous ossification of the mesenchymal connective tissue. Mesenchymal connective tissue containing blood vessels erodes the subperiosteal bone to invade the empty cavities. Some mesenchymal cells change to osteogenic cells, which in turn give rise to osteoblasts. Osteoblasts arrange themselves on the surface of the calcified trabeculae, start to lay down bone matrix, and deposit calcium salts. Once they are imprisoned in the calcified matrix, they are changed into osteocytes. Both osteoblasts and osteocytes form a regular bony trabeculae. This is immature type of bone. Some mesenchymal cells change to osteoclasts, while others change to bone marrow cells. Finally, all the calcified cartilages and their bony coverings are resorbed by the action of osteoclasts, with increase in marrow cavities. Thus, the embryonic bone formed in this way is immature. This is replaced by mature cancellous bone, which is followed by internal reconstruction or remodeling of such cancellous bone to form compact bone by continuous deposition of new bony lamellae on the walls of the marrow spaces, which gradually become narrowed to form the future Haversian canals and future Haversian systems. Factors affecting bone growth or ossification. Nutrition. Good nutrition is essential for healthy bone formation. For example, protein is very important for synthesis of collagen by osteoblasts. Vitamins also play an important role in bone growth. Vitamin A accelerates ossification and calcification. Its deficiency causes osteomalacia, that is, deficient calcification. Vitamin C is important for collagen synthesis. Vitamin D is very important for absorption of calcium by small intestine. Minerals also play an important role. For example, calcium is very important for the process of calcification. Hormones. Several hormones are needed for healthy bone formation. For example, parathyroid hormone increases blood calcium through activation and increase of the number of osteoclasts, thus promoting resorption of bone matrix and liberation of calcium in the blood stream. On the other hand, calcitonin hormone of parafollicular cells of the thyroid gland lowers blood calcium level through inhibition of osteoclasts and matrix resorption. Thus, it antagonizes the action of the parathyroid hormone. Both hormones together control the level of blood calcium and the process of ossification and bone formation. Sex hormones are also important for healthy bone formation and growth. For example, androgens of males and estrogens of females are stimulators of bone formation and accelerate the closure of epiphyses in adults. Growth hormone of the anterior pituitary also plays an important role during bone formation. During the growing years, an excess of growth hormone causes an increase in length of long bones, a condition which is called gigantism, while its lack causes a decrease in length of long bones, leading to dwarfism by acting on the epiphyseal cartilage plate. In adults, however, when the epiphyseal cartilage ossifies, an increase of growth hormone causes an increase in width of the bone only, a positional growth. Because of the lack of the epiphyseal cartilage, this leads to acromegaly. Joints. General characteristics. Joints are sites of contact between bones that may or may not allow movements. They can be classified either according to the range of movements they permit or according to the tissues that lie between the bony ends. Classifications. Fibrous joints, where a dense connective tissue joins the bones together, as in the skull sutures. Such joints permit a certain amount of movements during growth. Cartilaginous joints, where hyaline cartilage joins the bones together. These are either primary, permit no movements, as in the epiphyseal plates of growing bones between epiphyses and diaphyses, or secondary, which withstand compression forces and permit some movements, as in the intervertebral joints or discs and pubic symphysis. Synovial joints, on the other hand, permit free movements between the ends of bones, as in the knee joint or the elbow joint. Structure. By light microscope, the opposing bony surfaces are covered by articular cartilages and held together by articular capsule, formed of dense irregular connective tissue continuous with the periosteum. A complete or partial articular disc called meniscus may be present between the bony surfaces and is connected at the periphery with the capsule. The inner surface of the capsule is lined by a synovial membrane, which is a highly vascular connective tissue covered by squamous or cubical synovial cells, which secrete the synovial fluid. Synovial fluid is viscous, colorless, ultrafiltrate of blood plasma plus added mucin, rich in hyaluronic acid. Synovial fluid is an important route for nutrition to deeper chondrocytes of the neighboring articular cartilages. By electron microscope, synovial membrane contains two types of cells. A cells resemble macrophages and B cells resemble fibroblasts.